Great.
Yeah, so good morning, everybody. Thank you very much for attending the lecture number 9 in our series on risk assessment. So you may realize that now we are opening the section number 3 on emerging risk. And today we have a special lectures from Miss Hannah Gray from Johns Hopkins University, and she's going to talk to us about a topic that is very interesting and very of importance. Which is the occurrence of antimicrobial resistant genes in aquatic system. So before we're gonna learn more about the topic, let me spend some time talking about the profile of Miss Hannah Gray. So actually, she's a PhD candidate in the research group of Professor Bouwer and Professor Charles Young from Johns Hopkins University, and she's graduated from the University of Southern California with a bachelor and master in the same years, 2012. It's very interesting. And her research interests include, of course, antimicrobial resistance in wastewater and the environment. She's working as well on bacterial resistance, so I'm very curious to learn more about that. And she's working on novel detection methods and the resistance in diverse bacterial communities. So I just turn back the stage to you, Ms. Hannah, and thank you very much again for accepting giving these lectures to us.
Thank you. Thank you, everyone, for being here. I know it's probably kind of a weird hour for some people, so I really appreciate everyone showing up for this. And so, get started. So thanks for that introduction. Apologies. So, as you mentioned, my talk is titled The Occurrence of Antimicrobial Resistance Genes in Aquatic Systems. And so I'm going to ask you to actually kind of think more broadly about this, because genes are kind of an insufficient way to encompass all of the things we observe in aquatic systems. And so I want to talk about resistance as a whole, not merely just the genes themselves. So I'll start out talking about antimicrobial resistance, resistance in, excuse me for 1 second, what I'm viewing is coming up a little weird, so I'm just gonna change. Okay. Can everyone see my slides okay?
Yes, but could you please swing the slideshow into a presenter mode? So just swapping, yeah.
Okay, is that all right?
Yeah, it's perfect. Thank you very much.
Okay, great. Sorry about that. So to start out, I'm going to talk about the impacts of antimicrobial resistance. This is a hot topic right now. I'm sure everyone has heard quite a bit about this, especially in the news. I have a couple of articles pulled up here. One of them from November 28th says, guess what? They found antibiotic-resistant bacteria on the International Space Station. This actually doesn't seem like a particularly surprising thing, because in our guts, we are constantly carrying lots of antibiotic-resistant bacteria. There's also an article from about 3 years ago that reports that bacteria that resist the quote-unquote last antibiotic were found in the UK. And since that point, the antibiotic they're referring to is colistin, and since that point, we actually see huge numbers of incidents of colistin resistance, and I'll talk a little bit about that later. There's another one that mentions your gym mats may be breeding antibiotic-resistant germs, and this is a CDC image on the very right that talks about the threat level of methicillin-resistant Staphylococcus aureus, which we all know as MRSA. This is actually responsible for 11,000 deaths per year in the United States, as well as 80,000 resistant infections. So this is a serious issue that seems to be impacting all areas of our lives. As far as the impacts on public health, we see major impacts on food supply because of these resistant organisms getting into our meats and contaminating our food supply. Huge increase in healthcare costs because we have a reduced ability to both fight infection and limited or worsened clinical outcomes. As far as the impact on the environment, which is probably something we don't think about as much with antibiotic resistance, we see huge changes in the microbial community structure, which means reduced bacterial diversity in a community, which changes the stability of a bacterial system in the environment. And so this can mean that some biochemical processes that are really important can be interrupted due to resistance and the spread of resistance. And we're especially worried about this in wastewater because we depend on these important biochemical processes in order to adequately treat our wastewater. And so this actually creates a pretty bad cycle. Here are some statistics from the 2018 Antimicrobial Resistance Benchmark that was held at the World Economic Forum in Davos this year. And they report that there are almost 1 million deaths annually resulting from AMR across the world, and about 23,000 of these are in the United States. There are 741 antimicrobial medications currently on the market, and about half of these are specific to bacterial infections. We see 268 of these antimicrobial medications on the World Health Organization model list of essential medicines. So these are medicines that are crucial to global public health. One of the things that comes up over and over again is that in a lot of countries around the world, people lack regular access to antibiotics and antimicrobials. And it's actually this lack of access that can increase inappropriate use of antibiotics. So when limited access occurs, people might use antibiotics whenever they're able to get access to them, might not complete their course of antibiotics, or might be using an improper antibiotic for the infection that they have. So let's talk a little bit about where does AMR, which is antimicrobial resistance, and I'll refer to it as AMR from now on, where does it come from? So we like to break this up into 3 categories. The first is intrinsic, which is prior to the mass use and mass distribution of antimicrobials. And I don't say prior to the advent of antimicrobials because we have actually been seeing bacteria produce antimicrobials and other microorganisms produce antimicrobials to interact with each other. Apologize for the typo there. And an example of this is strengthened cell walls. We also see developed resistance, which occurs with mutation. And this is due to exposure to antibiotics, improper use, and other exposures to antibiotics other than merely taking them from a therapeutic standpoint, so low levels in drinking water. The last is transferred, which is probably the category of utmost concern, because transferred requires the transference of genes from one bacteria to another, either via transduction, which is phage-mediated, transformation, which is uptake from the environment, and conjugation, which is direct viable cell to viable cell gene transfer. And so we see this most often plasmid-mediated resistance, but transferred resistance means that you can have a non-pathogenic organism that's capable of distributing its resistance genes to pathogens. One of the most important things to note whenever we talk about where AMR comes from, that in the presence of antimicrobials in sufficient concentrations, resistance will be selected for. And so this means when we see antibiotics in wastewater, in drinking water, in the environment, we are constantly selecting for resistance. So we like to break down bacterial resistance into 4 fundamental mechanisms. The first being enzymatic degradation, which is the ability of a bacteria to actually release compounds that degrade an antibiotic. The second is alteration of targets. So if the antibiotic targets a specific enzyme, changing its conformation slightly can result in resistance. Bypass is one of the most crucial ones, which can be a bacteria changing its permeability, reducing the target site that an antibiotic might attach to, and therefore bypassing the mechanism of antibiotic action. The last, which is probably the most important for my area of research, is reducing exposure, which is efflux. So the ability of a cell to pump antibiotics out of the cell or away from the cell. And efflux is of a special concern because efflux can be nonspecific and therefore active on many antibiotics simultaneously. So we have a couple of different methods currently in use for detecting antibiotic resistance. One of the most classic ones is looking for the minimum inhibitory concentration or doing a phenotypic assessment of resistance. We usually have to do this on a single bacterial isolate, which requires an isolation step, most often based on species, on a species level. And we can create a bacterial lawn and employ the Kirby-Bouwer disc diffusion method, where we put discs that contain a certain concentration of antibiotic onto a bacterial lawn, and we look for a zone of inhibition, or a zone where the concentration of antibiotic is too high in the media for bacteria to grow in this. On the left, you can see that we have 3 different rings. The green ring, we see a substantial zone of inhibition. And so we can measure this zone and relate that back to whether or not the bacteria is resistant or susceptible. So when we have a big ring, this is usually a susceptible organism. And when we have a medium-sized ring like that in yellow, this is intermediate resistance, where there is resistance present, but there's also susceptibility to really high concentrations of the antibiotic, but outside a range of clinical relevance. And then the red is pure resistance. The bacteria can grow right up to the edge of that antibiotic-containing disc. The major limitations of this method, however, is that it takes a while, it requires multiple isolation steps and multiple growth steps. And so if the organism is slower growing, this can take a really long time and is not necessarily that useful in a clinical setting where you'll have already begun a course of treatment. This also changes between true conditions and lab conditions. So the way antibiotics might act in a human body can really differ from what we see on a plate. This also doesn't give us information about the mechanism of resistance. It just tells us whether or not resistance is present. And obviously this can be incredibly difficult when we're talking about environmental organisms, and impossible if we're talking about non-culturable organisms. So a lot of people have moved towards using genetic-based tests, specifically PCR assays, for specific known resistance genes, where we amplify a portion of the genome and then we can sequence these or use qPCR to get kind of a qualitative and quantitative measure, so both positive and negative, and a relative concentration of the gene. However, this requires specific knowledge of the genes and organisms. So if we don't have primers that we can use to target a certain gene, PCR won't be very useful to us. We also can't determine the activity or extent of resistance, which means that just because the gene is present does not necessarily mean that the phenotype is present. And University of Michigan is actually doing some interesting work around that topic. Also, not all resistance is due to a single gene mutation. And in fact, we see that very little resistance is due to that. And so it might require a huge amount of PCR in order to actually confirm that resistance is present. Of known resistance genes, there have been 20,000, give or take, potential resistance genes identified, and hundreds of confirmed resistance genes known. And there are a lot of databases out there that we can draw from that include these genes and various conformations of these genes. So this is just to give you an idea for how diverse a single phenotype can be. These are different genes responsible for quinolone resistance in aquatic environments in China. And you can see, based on the genus of these bacteria that exhibit resistance, we have a wide number of genes that can be responsible for that. So that can be very hard to do PCR for all of those. So my main area of focus is looking at the cross-resistance between broad spectrum, non-specific, non-therapeutic antimicrobials or antiseptics, and resistance to clinically important, clinically relevant antibiotics. Because of these various mechanisms of resistance and redundancy of genes, it's really difficult to attribute a single gene to antimicrobial resistance. When we have these antimicrobials, these non-therapeutic compounds, they often have different mechanisms at different concentrations, and so it can be really specific or it can be really nonspecific and hard to target a single gene. And multiple mechanisms of resistance allow for cross-resistance to occur, as I mentioned, specifically efflux. The two antimicrobials that I am particularly interested in are triclosan and benzalkonium chloride. Triclosan has a lot of name recognition. It is a broad-spectrum kind of antiseptic compound that was banned from consumer soaps and hand washes by the US FDA in 2016 after concerns arose about multi-drug resistance. It was banned from healthcare antiseptics in the US in 2017 and also banned from biocidal products in Europe in 2017. Canada is still considering a ban, but it also has widespread use in other consumer products. It's widely used as a preservative in a lot of personal care products and is in many surfaces that target or market themselves as antimicrobial. And there's significant presence in wastewater in the environment, as well as significant use in many developing countries. With triclosan, we note that gram-positive bacteria are much more susceptible than gram-negative bacteria, but its resistance from both positive and negative bacteria has been observed. The other compound that I'm specifically interested in is benzalkonium chloride, which is a representative of the quaternary ammonium compound group. These quaternary ammonium compounds are ionic compounds, and we often consider them the next generation of broad-spectrum antimicrobial antiseptic compounds. They have been in use since the 50s, but they're rapidly replacing triclosan in many consumer products, and they have also been detectable in wastewater. And I'll use BC as a representative. So on the bottom, you can see a couple of different products that are fairly well used that contain either triclosan or benzalkonium chloride. A lot of eye drops still contain triclosan in the US. If you've used soft soap, you have definitely used benzalkonium chloride. And the two boxes on the right are actually from a small market in the Amazonian region of Peru. And one of these is a soap that contains triclosan in the red box, and the other is a surface cleaner that says the materia activa is chloruro de benzalkonium chloride. So I specifically look at wastewater. One of the reasons we might ask why wastewater. One of the reasons for me is that I'm an environmental engineer. It's a system that's very interesting to me, but wastewater is also considered a genetic reactor. There are lots of interesting genetic processes taking place in wastewater. This is due to the very high bacterial diversity present in wastewater, and because of all of these unique substrates that are in wastewater and the high nutrient concentrations, we see rapid bacterial growth and very high bacterial concentrations. As a result of all of these substrates, we have very unique biochemical processes and a lot of population interconnectivity. We also happen to see very high concentrations of antibiotic and antimicrobial compounds, making this a very important system when we consider antibiotic resistance. We also see a lot of common wastewater organisms that have some low-level intrinsic resistance profiles that can very easily be worsened. These include some Pseudomonas species and Serratia species that I will mention again. So I'm going to frame this talk in terms of 4 case studies that look at exposure in terms of environmental reservoirs of resistance by characterizing antimicrobial and drug resistance and multi-drug resistance in these systems. So when we talk about environmental reservoirs of resistance, we're looking at systems that have a constant load of antibiotic resistance and antibiotic resistance genes. As a result, human exposure can result in repeated infection. Here's a little network that shows the relationship between humans and surface water and AMR in between that. We see humans bringing resistance in and out of hospitals, resistance going from humans into wastewater treatment, the bottom right, We also can see that industry can contribute to antibiotics going into the environment and wastewater systems. And then on the very left, we see a poultry operation where antibiotics are being used, and runoff from this into surface waters can result in AMR genes and AMR in surface waters. And we see constant interaction between humans and the environment and potential acquisition of these resistant organisms or genes. So the first case study I'm going to talk about is looking at the critical wastewater interface between humans and the environment. We can see wastewater treatment as almost a barrier to human exposure to both wastewater, pathogens, and AMR. And so because we have this wastewater treatment plant that's receiving effluent from the human population, from hospitals, and from industry, we're hopefully reducing the load of AMR that's entering into the environment. So I'm specifically looking at Back River Wastewater Treatment Plant in Baltimore, Maryland. And as I mentioned, wastewater is a really, really interesting system for looking at AMR. The second two case studies I'm looking at relate to extreme storm events. Because of these extreme storm events and aging infrastructure, we can have major overflows of wastewater and major leaks, which increase human exposure to wastewater as well as animal waste. So we see lots more runoff from animal systems, and then we see almost this wastewater barrier being bypassed. Looking at this in two systems, the first is in the Chesapeake Bay, and the second is in Hurricane Florence floodwater in North Carolina. The last case study I'll be talking about is a system where the wastewater treatment element is completely removed. This is in a community called Santa Clara outside of Iquitos in Peru. This is in the Amazonian region of Peru, and Iquitos is the largest city on earth not accessible by road. Because there's no wastewater treatment present at all, there's a constant exposure to wastewater and animal waste. So I'm going to talk a little bit about the methods that I employ for all of these projects. I specifically focus on doing a combination between conventional culture methods and sequencing. Oftentimes when we're culturing resistant organisms, we're targeting a specific species of organism and then looking at resistance within that population. I am actually starting with using these broad spectrum antimicrobials as a proxy for multi-drug resistance, as a way to specifically target organisms that have really extensive resistance profiles. And so I'm using both triclosan and benzalkonium chloride. I have plates that either contain 50 milligrams per liter triclosan, which is right around the solubility limit, or 250 milligrams per liter benzalkonium chloride, which is kind of the high-end concentration in consumer products. We do a multiple step isolation process and collect around up to 12 isolates from each media type, depending on how many colonies we see present. Then we perform Kirby-Bouwer diffusion, and then we've been sequencing many of these isolates and trying to gain as much information that relates phenotype to genotype. There's also been a side project where I've been looking at the microbiome of a lot of these systems that I'm talking about, but I won't be speaking about that today. Ooh, excuse me. I think I have music that just came on. My apologies. Okay. And as far as our sequencing and data analysis, we use sample prep with the Nextera XT kit, and we sequence on an aluminum iSeq at the Applied Physics Lab. We classify our sequences using MASH and assemble them with SPADE, and we look for genes with the Comprehensive Antibiotic Resistance Database and their resistance gene identifier, or GI. So looking at case study number 1, Back River Wastewater Treatment Plant receives water from roughly 1.3 million people in the Baltimore area, including Johns Hopkins Hospital. I have been sampling 1 liter of primary effluent from Back River 1 to 2 times monthly from January 2016 sorry, that's June of 2018 actually. And so this has been a really extensive long-term project. We've been doing DNA extraction from raw primary effluent as well as our TCS and BC plating protocol. Thus far, we have 314 isolates, and 183 of these have been sequenced. 75.5% of our strains that we collected came from triclosan-containing plates, while only 24.5% of our strains have been isolated using benzalkonium chloride, which might make sense given that benzalkonium chloride is just starting to be used more extensively. So just to look a little bit at the loading of resistance over time in wastewater, we saw, this is from February 2017 to December 2017, a fairly consistent concentration of culturable triclosan cells, ranging at about 10 to the 4 CFUs per mil, with our overall number of culturable organisms at between 10 to the 6th and 10 to the 7th cells, or CFUs per mil. Previously published benzalkonium chloride ranges, or MICs, range from about 100 milligrams to 562, and so we tested some different benzalkonium chloride concentrations. We settled on this kind of upper middle value of 250 milligrams per liter. And about at that range, we see about 10 to the 3 CFUs per mil resistant to BC, so about 1 order of magnitude fewer than triclosan. So we can start to look at what this cross-resistance looks like among all of these isolates. We have resistance profiles for these, and we can look at the percentage that's resistant, has intermediate resistance, and is susceptible to a number of clinically important antibiotics. One of the first things that's really noticeable is some of these antibiotics that are widely used, they have name recognition, like clindamycin, ampicillin, nitrofurantoin, which is used to treat UTIs. It's kind of the first antibiotic that people go for that. These all have extraordinarily high percentages of resistance in this bacterial library. We see a couple of antibiotics that have really low levels of resistance. Colistin and gentamicin, all the way on the right, are what we consider these last resort antibiotics. And colistin, as I mentioned, would come up again. About 15% of our isolates have complete colistin resistance. So this is a growing problem all around the world. Ciprofloxacin, which is pretty widely used, especially for just common we see a really, really low level of ciprofloxacin resistance, which is comforting. And then we have a lot of these antibiotics that have intermediate levels of resistance. Tetracycline, which is really widely used in poultry operations, is hovering around 60% resistance. And chloramphenicol, which is actually no longer used in humans, but is widely used in veterinary practices, is around the same level of resistance. So comparing these resistances between what's isolated on triclosan and what's isolated on benzalkonium chloride, we can see that for the most part, these are pretty similar. So even though these two compounds have really different mechanisms of resistance, triclosan specifically targets fatty acid synthesis, while benzalkonium chloride disrupts the membrane, we actually can see that a lot of these resistance profiles look really similar. We only have a couple of antibiotics where we see a really big discrepancy. Tetracycline in particular, we see a much higher level of tetracycline resistance when we have triclosan as our isolation mechanism, as well as with chloramphenicol. And with benzalkonium chloride, we see much higher levels of colistin and ciprofloxacin resistance. Interestingly, the mechanisms of tetracycline, chloramphenicol, colistin, and ciprofloxacin all differ from one another. Tetracycline and chloramphenicol both interrupt protein synthesis, though with different mechanisms. Ciprofloxacin disrupts cell division and cell replication by targeting DNA gyrase, and colistin is a membrane disruptor that targets phospholipids on the membrane. So I'm really curious as to why we're seeing these particular correlations despite these differing mechanisms of resistance. We also are able to, through sequencing, identify what the species present are. We see Pseudomonas come up a lot. We see lots and lots of Pseudomonas species, including Pseudomonas aeruginosa and putida, which are both opportunistic pathogens with some low-level intrinsic resistance, which is probably very easily amplified in these systems with lots of antibiotics and lots of resistance genes. We also see quite a bit of Serratia species, which once again are opportunistic pathogens with intrinsic colistin resistance. And we see a number of other pathogenic species present. 74.4 of our sequence BR isolates are clinically relevant and have been identified as infectious, which is, a particularly concerning statistic due to these extensive multi-drug resistant profiles. We also found 232 unique resistance genes using RGI among all of these species, and 43 unique species in our Back River isolates so far. And 43 unique species is actually not that high of a number considering the diversity of a bacterial population in wastewater. And so this certainly gets at this question of whether or not using cultural methods is the best way that we can access resistance. However, Pseudomonas should be something that we focus on extensively. The CDC lists Pseudomonas on their serious antimicrobial resistance threats list. It's a list of about 20 specific species that cause high rates of infection and deaths in the US per year. And with these Pseudomonas infections, they're actually on the rise. We also have very few instances of cross-resistance between triclosan and benzalkonium chloride, despite these really similar resistance profiles. Another thing we observe is that multiple isolates express benzalkonium chloride resistance and triclosan resistance on the same sampling date. So this probably suggests that there's a lot of lateral gene transfer taking place in the system at particular times. So for example, on November 9th, we can see that 3 of 12 isolates actually have cross-resistance between both benzalkonium chloride and triclosan, which is fairly low given that only 8% of our tested isolates show both of those resistance. CAC genes, which I'll talk about a little further, code for quaternary ammonium ion resistance, and these CAC genes are non-specific efflux pumps, but surprisingly we don't see high incidence of these, and we identify these in some species where we have not seen phenotypic benzalkonium chloride resistance. So the second case study is looking at the Chesapeake Bay. At the Chesapeake Bay, we have kind of 2 distinct sides of Maryland on the bay. The eastern shore of Maryland on the right is impacted by poultry farm runoff, and the western shore is more impacted by wastewater overflows. So the blue dots are wastewater treatment plants, and the purple dots are concentrated animal feeding operations, or CAFOs. The red dots are sampling sites, and here are kind of 2 newspaper headlines from this past year that are 3 days apart and describe huge sewage overflows in Baltimore waterways during heavy storm events. So we weren't able to collect that many samples from the bay. We have a fairly small sample set of only 13 isolates, but we can observe in these samples is that we have much higher levels of cefotaxime, which is a 3rd-generation cephalosporin, so one of the newer antibiotics out there, and colistin resistance in bay isolates than in raw primary effluent. Also, 90% of our bay isolates come from sites with a greater wastewater impact than poultry impact. So these are sites on the western shore, which also have a lot more human recreation, including fishing. All of these are from dry sampling events as well. So this means we didn't as a major wastewater overflow. So any organism that is present that we're observing, so sorry, my iTunes keeps popping up. The organisms that we're observing are organisms that are present in environmental waters without the major influence of wastewater overflow. So sequencing some of these isolates, what we can observe is something that backs that up, which is we see a couple of species among these species that we sequenced that are not observed in wastewater, but they are known opportunistic pathogens. These are Morganella morgani and Stenotrophomonas multifilia. So these are environmental organisms that are getting these extreme resistance profiles, specifically resistance to these antimicrobials that are used widely in human settings, but not so much in animal settings, that are probably acquiring these resistance genes from other organisms that enter the environment from wastewater. We also see Pseudomonas once again. It seems to be a very popular organism in this study. In our third case study, we were looking at Hurricane Florence rapid response, funded by the Johns Hopkins Center for a Livable Future, CLF. With Hurricane Florence, we saw significant flooding all throughout eastern North Carolina in these regions of really high industrial hog operations. We had extensive contamination of floodwaters and rivers with both wastewater treatment plant overflows and animal wastes. So we did repeat sampling effort along both the Neuse River and the Cape Fear River in eastern North Carolina, trying to quantify fecal pollution, antibiotic resistance genes, and antimicrobial multi-drug resistance. And this was a small portion of a much larger project that was also looking at the presence of antibiotics, the presence of heavy metals, and the presence of some specific pathogens like Staph aureus and E. coli in these floodwaters. So here's a map of this region in North Carolina, and all of those orange dots are swine CAFOs. So you see really, really high density of hog operations. We did 9 sampling sites, 3 of which were these upriver sites. And amazingly, 72% of our Hurricane Florence isolates actually came from sites upstream of these hog impacted sites, but we're also downstream of areas with high population density that had significant wastewater treatment plant overflows during this storm event, particularly in the Research Triangle. And you can see this red circle at the bottom right is that region of our upstream where we saw 72% of these isolates come from. So here's just an image, aerial image of one of these overflows. These are some chicken and hog operations that were just completely inundated with water. This is one of our sampling sites. You can see the neighborhood on the left. Normally behind the neighborhood, there's the river, and these are waterfront properties. And now they have become properties that are in the water, and the river and the road have kind of become the same. Here is another image from this, this is the Neuse River, and it is completely swollen. You can see this tree partially underwater. And so we did 4 total sampling events, and we were surprised to find that we actually needed to concentrate our samples in order to be able to collect isolates. So we were seeing fewer than 1 colony forming unit per mil, which is in comparison to the 10 to the 4 colony forming units per mil that we ordinarily see in wastewater. In total, we were able to collect 58 isolates, including 2 isolates that are TCS and BC resistant Staphylococcus aureus, which is fairly rare given that TCS mostly targets gram-positives, of which staph is one. We're prepping these isolates for sequencing, but I also want to note that only 14.7% of these isolates were BC isolated. So this is actually a lower percentage than what we see in wastewater. So to compare what this resistance profile from these Hurricane Florence isolates are to what we normally see in wastewater, one of the things that's worth noting is that our colistin resistance is much higher in this floodwater. We see it at a 44% rather than that 15% that we see in wastewater. We also see much higher levels of cefotaxime resistance, and we see higher levels of azithromycin resistance and trimethoprim resistance in this floodwater. And so that's one of the things that I'm interested in, is why we're seeing these different resistance patterns despite seeing actual fewer resistant organisms. Our last case study is looking at Iquitos, Peru. This was funded by an IGERT small grant, and we were there for a month collecting organisms from 16 total sample sites, ranging from sewage water outside people's homes all the way through the tributaries and into the river that the wastewater was flowing to. We were able to collect 139 resistant isolates, and we also extracted DNA from 30 unique surface water samples, and we're doing some metagenomic analysis on these. But surprisingly, benzalkonium chloride accounted for 64.7% of all of our isolates. So we see this much higher rate of BC resistance. And on the left, you can just see some of those different interesting isolates on plates. When we look at our cross-resistance of our Iquitos isolates by which media they were grown on, either BC or TCS, we see some pretty big discrepancies between our resistance profiles, particularly on the right with colistin resistance, that's CL. We see 72% of our BC isolates resistant to colistin, with 20 of our TCS isolates resistant to colistin. So this is a huge jump in colistin resistance. One of the reasons that we think this might be the case is because colistin is widely used in Peru in animal operations. There's a lot less control about what antibiotics people have access to, and people are able to just go to the store and buy these last resort antibiotics to use in their farming operations. Once again, we were able to sequence these and identify some abundant species. And the ones in red are species that we weren't able to observe at any of our Northern Hemisphere sites, specifically Providentia alcalifaciens, which is associated with diarrhea in children and traveler's diarrhea. And then we also see Chromobacterium, and that is a pathogenic species that actually gave that nice purple color on those plates. So we want to consider AMR as a function of the source of AMR. As I mentioned before, 90% of our Chesapeake Bay isolates came from sites with a greater wastewater impact than animal impact, and 72% in our Florence floodwaters had that same pattern. However, in Iquitos, Peru, where you don't have that barrier at all, we see isolates recovered from all sites, from wastewater streams all the way into the river. And we can also look here at this chart, which shows Iquitos resistance versus BMD, which is Baltimore, Maryland, and that's our Baltimore wastewater. What we can observe is that, for the most part, we see fairly similar levels of susceptibility with some major exceptions. Tetracycline susceptibility is actually much higher in Iquitos, the solid green bar. And this is because tetracycline is widely used in poultry operations, or we theorize this is the case because it's widely used in poultry operations in the States, whereas colistin, which is the other blue circle, is much more widely used in animal agriculture in Iquitos. And even though we're not seeing direct animal wastewater into our wastewater treatment plant in the US, this is probably due to this environmental reservoir effect, where people are being exposed to this resistance, possibly through food, or possibly through surface waters, and then this is cycling back through. We're also able to use the sequencing to look at family diversity among our isolates, and one of the things that we can observe is that in Iquitos we actually saw much greater diversity among our isolates, where in Back River we see a lot of these Pseudomonas species over and over and over again. But in both cases, we see that our benzalkonium chloride resistance contains fewer Pseudomonas species than our triclosan-resistant organisms. But overall, gamma proteobacteria really dominate these, and the other are only gram-positive, which make up a tiny percent of our isolates. Overall, we see the usage of antimicrobials in Peru differ vastly from use in the US. Triclosan resistance is far more prevalent in the US than BC resistance, and this is probably due to the extensive historical use of triclosan. We also see that the exposure to AMR in Peru is much higher than in US surface waters, even those impacted by storm events. Resistant bacterial concentrations in Peru range from 10 to the 1 CFU per mil to 10 to the 3 CFU per mil in surface waters alone. And source really matters. Multi-drug resistance due to antimicrobial resistance is likely a function of human wastewater than animal sources, whereas in animal sources we might see one specific resistance profile. So this kind of gets at the question of what comes first, the chicken or the egg, or in our case, what comes first, antimicrobial resistance or multi-drug resistance. And it seems like antimicrobial resistance is something that contributes. My goodness, I'm so sorry about this. I don't know why this keeps happening. I apologize. But it seems that antimicrobial resistance probably comes first. So looking at this from a genetic perspective, and I'll hurry up because I know I only have about 5 minutes left, so we have time for questions. It's been observed many times that we have high diversity of resistance genes in wastewater and activated sludge. There was one project that identified over 200 subtypes of antibiotic resistance genes, or ARGs, in activated sludge, and another that demonstrated that there was very little degradation of ARGs in composted sewage sludge even after processing. So we know that there's a high concentration of these genes present. There's also been a lot of focus on integrons, which allow bacteria to capture and mobilize genes contained in mobile elements called gene cassettes. And specifically, class 1 integrons, we can identify these as potentially correlated to the presence of AMR gene cassettes, which code for multi-drug resistance. Our PCR-based detection is still really widely used, and we can use it for CL1s to look at multi-drug resistance particularly. In the isolates that we sampled, we see a couple of resistance genes appear over and over again. One is OQXB, which is a nonspecific efflux pump, usually plasmid-mediated. It's present in multiple different species that we've identified. Another is SOXR, which is an oxidative stress response superoxide activator, which once again appears over and over again, specifically in Pseudomonas species. But overall, our isolates range from having 0 known genes identified up to 41 genes identified, which just kind of reiterates the problem with using PCR alone as our way to determine whether or not a resistance phenotype is present. Overall, we had 262 hits for 50 known triclosan resistance genes. And in many of these isolates, they actually had all of these 15 known triclosan resistance genes. We only had 21 hits for 3 known benzalkonium chloride resistance genes. And this includes in our Peru isolates. And so we know that the phenotype is present, but these known genes are not. We also identified in total 273 unique resistance genes, 113 of which were efflux, which makes sense when we're talking about multi-drug resistance. Efflux can be nonspecific and therefore target many antibiotics at once. This is very complicated and kind of excessive chart, but what I really wanted to draw your attention to is the whole top row and most of the bottom row is all one isolate. And it's Pseudomonas aeruginosa, and you can see the presence of all of these genes, and all of the red Ys are if we actually observed the phenotype that these genes correspond to. On the right, the bottom corner, is a different Pseudomonas species, and we were only able to identify 2 genes despite this pretty extensive resistance profile. Of the genes that were identified in the top isolate, the majority of these are efflux pumps, and 5 triclosan-specific efflux pumps were present. But you can see that we can see a really wide range of the number of resistance genes despite what our phenotype might demonstrate. In a couple of our isolates, like our Archobacter bacilleri isolate, we identified no known resistance genes. We only identified a single resistance gene in one of our few gram-positive isolates, but in Pseudomonas aeruginosa, we know that there are a number of resistance genes present that we have been seeing over and over again. We also see a lot of membrane impermeability resistance genes in Pseudomonas, and specific beta-lactamases, which we expect to be present. But one of the things that I really want to reiterate is that the presence of these genes does not necessarily equal the expression of these genes, but the absence of these genes does not necessarily equal the lack of expression. As we saw that that one isolate that only had 2 genes present, or previously we identified an isolate that had a CAC resistance gene but did not see benzalkonium chloride resistance. So I want us to kind of reframe AMR as potentially a contaminant of emerging concern, instead of just focusing on the gene. And this is a little bit of an abstract notion, because genes are so difficult to detect, and it's not just a gene, but the expression of a gene or a gene combination that actually results in AMR. So looking for the genes as the contaminant themselves is not necessarily sufficient. The last thing that I'm going to spend just 1 or 2 minutes talking about is another aspect of my project, which is using microfluidic droplets to access more environmental diversity to get a better idea for what the total resistome is. Because of this great plate count anomaly, or our inability to culture most of the environmental organisms, we see a very limited genetic set of what the resistance is. Also, the genetic methods for antimicrobial resistance are limited, as I mentioned. And so one of the things we've been trying to do is encapsulate these environmental organisms in droplets with a single cell inoculant, which means that we don't have to introduce culture media, we reduce competition between fast and slow growing organisms, and we have really high throughput, which is much higher than what we can do with plates, which means that we are more likely to access rare organisms. So we're trying to employ that in order to get better access to these organisms. And so I'm happy to answer questions about that too. So a thank you, of course, to Ed Bouwer and Chuck Young, my advisors, the Bouwer Lab Group, Kathleen, who's helped me enormously with sequencing, and Keith, who's helped me with sequence analysis, our Hurricane Florence team, and our Iquitos team, and funding sources from the NSF and the GRFP. And now I'll take any questions. And this is a view of the Amazon River from Iquitos.
Awesome. Thank you very much, Ms. Gray, for this very interesting talk. Very field-based and it's very top research. We may have some time for some questions. I just wonder if the audience may have some. So you could even speak it out, or you can just type the question in. So I see here a first question from Professor Evans, who thank you for this very interesting talk. And the question is...
Oh my goodness, there's that music in there. I'm so sorry. Apologies. I don't know why that keeps happening. Okay.
Yeah, so the question is, there is any difference in environmental persistence or viability of resistant bacteria compared to non-resistant, particularly for those that are resistant to last resort antimicrobials?
That's a very interesting question. Thank you for that. This is something that I actually haven't really focused on, but I do think that that is something that we might be able to observe with those Chesapeake Bay isolates, and with me delving a little bit more into the microbiomes that we see in these environmental waters. I would theorize that we do see increased environmental persistence of these resistant organisms, largely because we have a significant influx of antibiotics into a lot of these environmental systems. There has been some research that shows that tributaries that are impacted by treated wastewater have a much lower diversity than what you would normally expect in an environmental tributary, much lower bacterial diversity, which might suggest that we have persistence of these resistant bacteria and die-off of these normal environmental organisms.
Awesome.
Thank you.
Thank you. There's any other questions? I may jump in with one of mine. So we know the source matter, right? So
yes.
There's anything that we could do to be able to limit and confine this high activity of the number of bacteria growth and the number of genes to be confined in the area around the wastewater treatment only?
Yeah, I mean, I definitely think that improving our treatment processes is probably going to be one of the most important things in order to control both AMR and these ARGs, these resistant genes. Specifically utilizing processes like potential UV paired with oxidation in order to break up some of these genes and prevent them from entering the environment. Another thing that we've certainly looked at is eliminating these overflows. Trying to prevent this barrier from being broken would vastly reduce the amount of resistance that we see entering into the environment.
Yeah. And another question is, just to confirm, please, if I didn't understand it wrong. So colistin is one of the last antibiotic for human health, right? And
yes.
why that would be like used for veterinary protection in Peru?
So there was a lot of thought for a long time that there wasn't so much crossover between resistance in animals and resistance in humans. In humans, we don't use colistin very much because it is extremely hazardous to human health. However, in a lot of animal feeding operations, antibiotics are used much more extensively. They're used prophylactically, so when we don't see an infection present, they're still employed. And so there are much higher rates of resistance to some of the more common antibiotics in these animal operations. And so they've been the first group to really jump towards using these last resort antibiotics. But the thought was that there wasn't that much overlap, and we see, obviously, that's not true.
Awesome. Thank you very much. I just wonder if there is any other question from the audience. Awesome.
Great.
Yeah, thank you very much again for this great talk. And thank you very much for the attendance to the series. So you may realize that we're going to have a break after these presentations. We are going to have the 2 other lectures again in January. And so I just want to wish you a warm and nice Christmas. And then so Merry Christmas and Happy New Year to everybody. Thank you.
Thank you.
Bye-bye.
Bye.